Very happy to be here today. So thank you very much to the organizers of this webinar. It's been very interesting so far. Amazing talks about zebrafish, the model, how to improve the welfare of the fish, and also like the imaging techniques. A really interesting talk. And yeah, so as you've said, my name is Ana Hernando, and I'm COO and co-founder at Bionomous. And I will explain to you the EggSorter, which is an automated tool to standardize the screening, sorting, and dispensing of zebrafish embryos. So, as you might all be aware of course, the zebrafish embryo model is a widely used model in research because it has many different advantages. So, to say the high fecundity of the fish, the fact that the embryos are a whole organism, that they develop externally and that they develop fast, that they are transparent, and that there are like many genetic and molecular tools to use with this model, as well as they allow for high throughput assays. Besides, the zebrafish embryo model is an accepted alternative model to animal testing in other vertebrates. However, not all that shines it's gold. Well, even if this case it's almost gold, because there is still some problems associated with the zebrafish embryo model. So one of the problems is like one of the advantages is like they are like yeah, it's the high fecundity, so we have a lot of embryos, but these embryos are quite time consuming to process and quite tedious. And besides, there is a problem with the lack of experimental protocol standardization, which might lead to the lack of reproducibility when using this model. And this is when Bionomous wants to play a role. So we have developed the EggSorter, which is an automated tool for the screening, sorting, and dispensing of zebrafish embryos. So we want to standardize the zebrafish embryo processing. And you may wonder how and how does it work? So I think I can put now, yeah. So the EggSorter has an easy entry for the samples. So there will be one input, and then the samples will be guided to a wheel, to this sorter, that has small slots that will take the eggs one by one, and that will place the eggs in front of a camera. Then the camera will take a picture of each of the eggs, either in bright field or in fluorescence, and these pictures will be classified by using artificial intelligence. Then, based on the classification of the images, the eggs will be sorted out into different containers. So to say, for example, we want to discard the bad eggs into falcon tubes, and we want to plate the fertile eggs into multi-well plates for the experiments. And so for you to know, the dispensing can actually be done in not only in multi-well plates, and in multi-well plates it can be done from 6 wells to 96 wells, so 6, 12, 24, 48 wells, and you can choose its position independently, so it doesn't have to be the same. So we have 4 different positions, and you can have multi-well plates as well as petri dishes. And what is cool is that you can also choose the volume of medium per entity, and the medium can either be like system water or embryo medium or any other salty medium that you might be using. And you can choose the number of entities to plate per well or per container. And a really cool feature that we just developed is this compound dispenser. So we will automate not only the processing of the zebrafish embryos, but also the dispensing of the compound of your interest directly with the eggs. So for example, if you're using a multi-well plate, you can dispense directly in the well plates the drug of your interest and select the volume of drug you want to dispense, as well as in which multi-well plate you would like to plate it. And as it might be much easier to just see how it works with a video, I would like to play this video for you. So because it's much better than a lot of words. So you can see here in the video the current protocol. So it's a researcher with a pipette and a microscope screening by hand the entities and plating them and taking notes manually of what they're doing. So with the EggSorter, what we want is like to automate this process. And as I've mentioned before, you see there is this one entry for the eggs where you can pour directly the eggs, and then the eggs will pass to this wheel that is grabbing them one by one. Then you can see in this screen, which is actually a touch screen that is controlling the whole device, the pictures in bright field and inflorescence that the EggSorter can take. And then now you see the dispensing that the device can do. And here there's like a small and funny comparison. Of course, no drinking of coffee in the lab, but as you see, the researcher that is doing it manually can only do the sorting, while the researcher with the EggSorter can do all the other tasks that there are to do in a lab. So when we compare then the benefits of the EggSorter, it releases 80% of the time with 95 to 99% accuracy in a fully automated process. So now that you've seen how the EggSorter works with the video, I would like to go into detail into some of its features. So one of them being the artificial intelligence algorithms. So as you can see in the screen, we have an algorithm embedded in the EggSorter that it's able to recognize between fertilized and unfertilized or dead embryos at different developmental stages. So when considering all the different developmental stages together, we have an accuracy of almost 95%. And when we go stage by stage, the accuracy of our algorithm is always over 90%. And here are some images of eggs inside of the EggSorter, fertilized ones at different stages of development, as well as unfertilized ones and dead ones. And as I've said, the EggSorter cannot only take pictures in bright field, but also in fluorescence. So for the fluorescence detection in the EggSorter, there's a space for up to 6 different wavelengths. So we have channels for blue, we have channels for green, for yellow, and many different channels for red. And it can also detect multifluorescence, meaning that imagine you have a line with 2 different markers, so red and green. You will be able to select the double positives and place them in one multi-well plate, or to select the single positives and place them in another Petri dish, for example, or in another multi-well plate. And the classification for the fluorescence is intensity-based, meaning that we have 2 different thresholds in the EggSorter, one for the intensity and one for the area, that can be combined together to detect the fluorescence that you're interested in. And then, as you might wonder if the EggSorter, whether the EggSorter has an effect or not in the development of the zebrafish embryos. So we've run different tests to check how the embryos survive after going through the EggSorter. So in this case, we processed the eggs in the EggSorter at 4 to 8 hours post-fertilization. And then we compared the health status with a control, which means like the eggs that did not go through the device. And as you can see in the graph, so in light blue, it's the results for the eggs that were processed in the EggSorter, and in dark blue, it's the control. There are no significant effects in the health status of the embryos after 5 days post-fertilization. And just I would like to go through this use case because it's always easier to imagine how this technology could be applied to standardize the protocols for zebrafish studies using the zebrafish embryo model. So by this has been following the OECD guidelines. So in the current protocol, when we test 5 compounds, we will need to produce like actually the double of eggs that are needed for the experiments, because the protocol takes into consideration that there might be some errors that might arise or some things that might not work. And it takes around 4 hours of manual screening and plating. And besides, all the data has to be taken manually, which unfortunately might be error prone. While if we compare this with the use of the EggSorter, we will need to produce less eggs, as the margin for the error, it's already known and is less. It's a standardized selection of the eggs. And how it will work is like the user will need to interact 4 times for this amount of eggs with the machine, and then the machine will run for 3 hours automatically. Besides, once it is finished, the EggSorter will save all the data automatically and will create reports with the results of the experiment. So it will standardize the process, and it will save over 80% of the time and increase the efficiency by 40%. And just we don't stop with zebrafish, because the EggSorter can also standardize protocols with similar models, with other fish models. So we have used this technology with Medaka eggs, as well as for killifish eggs, which are two other models widely used for research or for toxicity studies. And what you can see in the pictures, it's like we cannot plate only in like Petri dish with water, but also with some solid media. So a bit in conclusion, it's that the zebrafish embryo model is a great alternative due to its many advantages and it's a great alternative to animal testing in other vertebrates. Refinement for zebrafish embryo testing will pass by standardizing and creating new protocols to work with this model. And automation is key to provide this standardization and help in the adaptation of the zebrafish embryo model and similar models. And I would like to give an overview on how this idea started and where we come from. So Bionomous is a Swiss company. So we started at the EPFL in Lausanne. Then we moved later on to the countryside. So actually in the Canton de Fribourg. So not really far from the University of Fribourg, from where one of the presenters is from. And then just for you to understand a bit how this technology evolved. So we started in 2017 with the idea, and then we iterated like different prototypes that were placed at clients. We had the feedback, and then last year we launched a commercial product that is available, and that is the product that I just presented. And just so that you know, we don't end up in the automation with the zebrafish eggs, we're also developing a larvae sorter together with the CSEM, which is a Swiss Research Institute. And it's the same concept as the EggSorter. So for you to understand, the EggSorter can work when the eggs are still in the chorion, while the larvae sorter will be the next steps when the eggs will hatch and will have the larvae. But the concept is the same in the sense that we will individualize the larvae. So here you have a picture of the picking of the larvae inside the first prototype of the larvae sorter. And then the larvae will be guided to a screening chamber where we can screen in bright field and in fluorescence. And then based on the classification of the algorithms, then the larvae will follow different paths. So with this, I would like to say that we're looking for early adopters of this first prototype of the larvae sorter. And also, if you have any ideas about this technology, please do not hesitate to reach out, or if you want to test it in-house and see how it works. And with this, I would like to finish the presentation by thanking you all for your attention. And please do not hesitate if you have any doubts.
Thank you very much, Anna. It was really insightful to learn about this device and how it can be used in the lab. Thanks a lot. Let's see if there are some questions from the audience. So feel free again to write either in the chat, the Q&A, or raise your hands.
Thank you very much, Anna, and congratulations for the development of this really, really great device. We have here some questions. So one is, until what stage can you use the EggSorter? Can you use it after hatching or on the second day when the chorion is softer and the fish already moves?
It's a very good question. So we cannot use the EggSorter once the larvae have hatched. And what we recommend is to use the EggSorter until like 40, maximum 48 hours post-fertilization, because it's true that at some point the chorion can get very fragile and then it will break inside the machine. So maximum 40, 48 hours.
Thank you very much. then you say that the sorting is not done with a 100% accuracy. Is this due to a mechanical matter, or is it due to the artificial intelligence? And are you expecting that this will improve?
It's a good question. So it's a bit of both factors. So there's a bit of mechanical, like I would say, what's the sense? Like it can happen that we lose maybe some eggs, but this is something we're improving, and it's not like it's really less than 5% of the eggs. And then it can also be like a software, but then the software we're currently improving it, and like what we want to reach is like 99% accuracy.
Okay, thank you very much. So one last question, I think. So what are the possibilities of contaminating a fish line by this automated process? So when you're working, is there a high risk of cross-contamination?
So normally what we have in the device, it's a pure eggs sequence that will make sure that there are no eggs left in the machine between one line and another line, so that we make sure that there are no eggs left. And otherwise, if you want to be 100% sure, you can always empty the device from water. This will remove all the eggs that are inside and refill the device with water, so that there are no eggs left from previous experiments, and the risk of line contamination decreases quite a lot.
Okay, great, thank you very much. So if there are people that are interested to be an early tester of the EggSorter, should they contact you directly?
Yes, please. I think there's my email in the slides, so please feel free to reach out.
Thank you.
Perfect, thank you.